Have you ever wondered how the food we eat every day turns into energy and nutrients that our body can use? This happens through a process called digestion, which is the process of mechanically and enzymatically breaking down food into substances for absorption into the bloodstream. A key part of chemical digestion is the action of enzymes. An enzyme is a protein with catalytic properties due to its power of specific activation. During digestion, enzymes break down large food molecules such as carbohydrates, proteins, and fats into simpler molecules like glucose, amino acids, and fatty acids. Without enzymes, digestion would be too slow to supply the body with essential nutrients, highlighting the vital role of enzymes in digestion.
Imagine biting into a juicy stick. Those proteins look harmless, but they're giant molecules too big for your cells. Enzymes are the macroscopic chefs, chopping them into bite-sized amino acids. So, large macromolecules like proteins, starches, and triglycerides in our everyday diet will be repelled from the thick phospholipid bilayer of the cell, as the bilayer's hydrophobic layer would repel polar charged macromolecules and its specific channels, and transporter only allows small and compatible solutes as well with their high membrane selectivity. Thus, these macromolecules would be rejected by the cell. Malnutrition will occur if no countermeasures were taken. This is where our enzyme comes in. Enzymes are extremely vital in digestion as they catalyze the hydrolysis of the macromolecules into monomers like amino acids, glucose, and fatty acids that the body is able to absorb more easily. For example, there are several specific digestive enzymes like salivary amylase in the mouth, pepsin in the stomach, and pancreatic amylase, protease, and lipase in small intestine to ensure complete nutrient utilization, supporting energy production, growth, immunity, and toxin removal by the liver. Without enzymes, you chew for days. Mechanical mixing alone can't break chemical bonds as chemical reactions occur too slowly at normal body conditions at 37 degrees Celsius. Starches would sit like rocks, causing bloating, malnutrition, and energy crashes.
How does enzyme work? The workings of an enzyme can be explained as far back in 1894, which is the lock and key model as the first proposed and the older model. The lock and key model shows that the active site of the enzyme has a specific shape and only the substrate with the right configuration can bind to its site, mimicking to how a real lock and key works. Only the right key can fit into their own specific lock. While for the newer version is the induced fit model where it's slightly different compared to the lock and key model. Here, the enzyme actually changes its shape here. How, you may wonder? The active site of the enzyme slightly alters and adjusts its conformation according to the complementary substrate. With that, the substrate is able to lock in place and start off the catalysis process. In the digestion process, enzymes apply the induced fit model to bind with substrate at active sites. This can lower activation energy needed by stabilizing their transition states and rapidly splitting bonds of the substrate without extreme heat and pressure and can still be used in the future. For example, cofactors like colipase and bile salts aid lipase in emulsifying fats into micelles for adsorption and increase surface area for lipase action. This process aligns with digestion stages. In the mouth, amylase hydrolyzes starches into maltose. In the stomach, pepsin cleaves internal peptide bonds of proteins to peptides in acidic pH. And in the small intestines, hormones like CCK, or what it stands for cholecystokinin, activates pancreatic enzymes at optimal and neutral pH. pH shifts, or inhibitors, would denature enzymes and halt catalytic activity, but cofactors like bile can aid lipase efficiency in fat digestion.
In conclusion, enzymes are essential biological catalysts that play a crucial role in the human digestive system. They allow digestion to occur efficiently by speeding up chemical reactions that break down complex food molecules into simpler form that body can absorb and use. Without digestive enzyme, nutrients such as carbohydrate, protein, and fat would not be properly digested, leading to poor nutrient absorption and reduced energy for daily activities. Digestive enzymes like amylase, protease, and lipase each have specific and important functions. Amylase begins the digestion of carbohydrate by breaking starch into simpler sugar. Protease break down protein into amino acids needed for growth and tissue repair. And lipase digests fat into fatty acid and glycerol for energy storage and cell function. These enzymes act at different stages of the digestive lineage, from the mouth to the small intestine, ensuring that digestion is carried out in an organized and effective manner. Overall, the coordinated actions of digestive enzymes ensuring that the body receives the nutrient it needs to maintain health, support growth, repair damaged tissue, and regulate bodily processes. Therefore, enzymes are not only important for digestion but also vital for the survival and proper functioning of the human body.